Hello, and welcome to my DNA replication power note. So the goal of this presentation is just to basically build upon the videos that you watched yesterday and see if I can't make things a little bit clearer and go over the functions of some of the enzymes that are involved in this process. So on your notes template, you've got a little bit of text on one side and a diagram. So we're going to start by annotating the text just a little bit by adding some highlights. So the process of DNA replication is very similar in both eukaryotes and prokaryotes, but we're going to focus on eukaryotes. So what I've done here is I've highlighted DNA synthesis and DNA replication just so you can kind of make a correlation between the two terms. Synthesis is just the replication of DNA. During the S stage of the cell cycle, DNA is loosely organized in the nucleus. Certain enzymes start to unzip the double helix at places called origins of replication. The double helix unzips in both directions along the strand. Eukaryotic chromosomes are incredibly long, so they have to have many origins of replication to help speed up the process. Other proteins hold the two strands apart, and we are going to talk about those enzymes in a little bit more detail later in this presentation. So the unzipping, and I did a bad thing here. This one right here should actually be right here. So if you'll make that change on yours, because it should read the unzipping has two effects. One, it exposes the bases on the DNA strand, and two, enables free-floating nucleotides to pair up with their complementary bases. So A's will bond with T's, C's with G's, etc. DNA polymerases bond the nucleotides together to form new strands that are complementary to the original template strand. Now, DNA polymerase is highlighted in a weird way because I want to make a comment on that. Those are enzymes. I know they're enzymes because they end in ASE. So the result of DNA synthesis is two identical strands of DNA. DNA replication is described as semi-conservative because each DNA molecule has one new strand. That's why I've got the star here. It's got a new strand and one original strand. So if you'll add those little asterisks right there, just to kind of draw your attention to it. Finally, DNA polymerase not only one bonds the nucleotides together, it also has two, a proofreading function. It can detect incorrectly paired nucleotides, clip them out, and replace them with correct nucleotides. This is pretty interesting. Uncorrected errors are limited to about 1 per 1 billion nucleotides, which is pretty good. So before we continue, I just want to remind you about DNA structure. We talk about how it's anti-parallel, so the two strands run in opposite directions. They actually run from this 5' to 3' direction here, and on the other side, 5' to 3' direction here. And so when we talk about DNA structure, the fives just refer to the number of carbons in the deoxyribose sugar. And scientists number them off, and that's neither here nor there. The easiest thing to do is when you're trying to figure out which is 5' and 3', is to look at the pentose or 5-carbon sugar. It is sort of shaped like a house, and I call it a sugar house in class. So the 5' end would be the pointy roof end of the deoxyribose sugar, and it's always going to have a dangling phosphate as well. And then conversely, if we look at the other side, we can see that the sugar is seemingly upside down because it runs from 5 to 3 in this direction. And so the 3' sugar is actually down here at the corner bottom part of the sugar house. And so really, DNA runs in 5 to 3 in either direction. So just the general description of DNA synthesis, it's just DNA is copied during the S part of interphase. And before we start on the diagram portion of this, I have this quick video to show you.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way.
So I want to pause it here just for a second to kind of don't get too bogged down in the leading lagging strand. The easiest thing to remember, and I'm going to repeat this in just a little bit, is that the replication DNA is being opened in this direction. Okay, it's being unzipped in this direction here, and so the leading strand is replicated
Way because it runs in the
in the direction of the replication fork. And so it's just super easy. Boom, 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 boom. We add those nucleotides, super easy to copy. The lagging strand is going opposite. And so that makes it a little more problematic. And so the sections have to be added. It has to be replicated in chunks. And so we call that the lagging strand.
in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, so on your diagram portion, you got the replication diagram right in the middle. Then you've got like the identify the structures on the right side of it and the process on the left side. So let's start with the right side numbers, and we're going to identify the things that those numbers are pointing to. So 1 is pointing to the sugar phosphate backbone of DNA. 2 is pointing to actually two things. One of those is a loose nucleotide. The other is like literally a nitrogen base hanging off the nucleotide. And then so I did NB for nitrogen base, so you can abbreviate it. 3 is pointing to the enzyme DNA polymerase, that big gray blob that's actually adding the loose nucleotides to the synthesizing strand. And 4 is the newly synthesized strand that's being produced. Now, as far as the process, here's where we're going to talk more about the enzymes in a little bit more detail. So these are the boxes that are on the left, the larger boxes, not the lined ones. So step 1, helicase is going to unwind DNA, creating a replication fork. Primase is going to kind of initiate replication at the origins of replication. So it's just going to tell all the enzymes involved, hey, this is where we're going to start this process. DNA polymerase is going to add loose nucleotides one by one in the 5' to 3' direction. So we're going to tack them on to the bottom of the sugar house, if you will. That's the direction in which they are added. I mean nucleotides. And then step 4, you get 2 molecules of DNA, each identical to the original. Each contains a new strand and an old strand. So, like I said, don't get too bogged down in the leading strand versus the lagging strand. It's really not difficult to tell if you just look closely at a diagram. So all you have to do is pay attention to the direction in which the DNA is unwinding and unzipping. So here it's unzipping in this direction, which means that the strand that's being synthesized in the direction of the replication fork, right? That's the easy strand, that's the leading strand. If it's being constructed in the opposite direction of the DNA unzipping, that's the lagging strand. So just a little bit more on number 3 in detail. I've actually put this in a text box for you there next to the diagram. But remember that DNA strands are anti-parallel. This is just a summary of everything I've said in one place. DNA strands are anti-parallel. Replication occurs continuously on the 5' to 3' strand. That's the leading. Occurs in segments along the 3 to 5 strand, which is the lagging strand. It has to be done in chunks. Those chunks are called Okazaki fragments, and they're sealed together by an enzyme called DNA ligase. So here's another diagram for you. And you can see this time it's like we took the previous diagram and we added more stuff to it and then we flipped it around. So now the direction of DNA unzipping is in this direction, okay, this way. And so you can see that if it's the strand that's being synthesized towards the replication fork, adding to the 3' end, that's the easy side, that's the leading strand. This side is going to be the lagging strand, and these sections are called Okazaki fragments. So last little bit, factors for accuracy. One of the things that keeps replication accurate is just the specificity of base pairing. A's can only bond with T's, C's only with G's. And then DNA polymerase does some proofreading. Now, what happens if DNA polymerase doesn't catch it? Okay. Well, first, you have this thing called mismatch repair, and that will remove or replace incorrect nucleotides, which are errors of replication. And then secondly, you have nucleotide excision repair, which is a fancy way of saying we're going to remove and replace incorrect nucleotides. That can be due to DNA damage, which is usually from environmental factors such as chemicals, X-rays, UV rays, and smoking. All right, that does it for me, and I hope you found this helpful.